Welcome to your lesson on biotechnology, the first part. Let's dive right in. Okay, so one thing I need you to understand is that the vast majority of the tools that we have, the biological tools that we have, we didn't invent. We've stolen them from living organisms. So the first few that we'll talk about here, if it's not a machine, like an actual machinery, we stole the idea, the enzyme or whatever, from a living organism, figured out they had it, stole the tools that they already use. So part of what you want to pay attention to is going to be what was the original purpose for that particular enzyme, let's say, and how are we using it now? So kind of focus on that as we go in. So the very first one that I want to jump into is called recombinant DNA. This is the one that was the idea that was first patented in the 70s, 1973, I believe. The one that they first started making money on. When they figured out how they could mix and match chunks of DNA to make a product, they suddenly had a use for DNA. So recombinant DNA is a very important technology. And there's a whole bunch of different pieces to the technology that we use to make the recombinant DNA. But the fancy idea is just to say, this DNA sequence from this particular organism, let's steal it and stick it into somebody else's DNA sequence. And then whatever is written on that code, the new organism will make the same thing. For example, we've done that with bacteria. It's way too labor intensive to try to suck insulin out of somebody's pancreas to give it to a diabetic. So instead, you take the human insulin gene, give it to bacteria, and then they happily produce all the insulin you'll ever need. You just siphon it off, and there you go. You've got human insulin created by a bacterium. So pretty clever. So that's the type of thing we've started using this for over the last 50 years, I should say. Okay, so recombinant DNA, how do we actually analyze and alter these genes? And again, we're always interested in the protein product because that's the visible expression of that gene. Okay? So the first tool, super important, is these restriction endonucleases. The idea of restriction just means that it's at a very specific code, set of letters in a row, exact set. So it's restricted to cut at that particular sequence. And the idea that it's an endonuclease just means it's inside doing its job. So it's an enzyme, an ASE enzyme, cutting nucleic acids, that's the nuclease part, and it's inside doing this. Okay, we've stolen these from bacterium. They're the ones that we got it from, so we name them after bacteria. But the basic thing is scissors. And they're going to cleave, that's the word cut, make sure you know that word. They're going to cleave a double-stranded DNA sequence. The short form for double-stranded DNA is dsDNA, as opposed to if you had a single strand. Most DNA is double-stranded. So it's going to look for a very specific set of base pairs, and that base pair set is called the recognition site. Depending on which scissors you have, they can recognize different sites, but they're usually somewhere between 4 to 8 base pairs along in the sequence. So BP for base pairs from here on out. So far we've discovered over 2,500 different enzymes that'll do this job for us. And between them, there's about 200 different sequences that they'll cut. That means that some of them overlap. They might cut the exact same sequence as another endonuclease. Okay. Now, the cool thing about these endonucleases and the recognition sites is that the actual site itself has a special characteristic. It's a palindrome. What's a palindrome? Forwards and backwards, exactly the same, right? You know any examples of palindromes? Race car is a good one. Taco cat, pretty common one. You get the idea that same forwards and backwards. Any other ones? Mom, wow, dad, level. We even have ones that are sentences. Level's a good one. We have ones that are sentences as well. There's a famous one, has to do with Napoleon Bonaparte when he lost and then was exiled to the island of Elba. So there was this the whole sentence is a palindrome. Able was I ere I saw Elba. So you can get them pretty long sometimes, right? But these ones are going to be 4 to 8 letters long. Same in both directions. And remember, when we're talking direction here, we're talking about 5' to 3'. And then when you flip down to the other piece of DNA, its 5' to 3' is the same. Okay? So how do we name these suckers? Well, they get named after the bacteria that we stole the tool from. So when you see the first one here in the example, BAMH1, it's not an I, but it's a Roman numeral 1. The B is from what genus it came from, so that's a bacillus, tells you the shape of the bacterium. The AM comes from the species name, Malik fascians, and then H is which version of that particular bacteria, so the strain, and then I is not really I, it's a 1, but this is the first one that they found. So there should be a BAMH1, BAMH2, BAMH3, however many they found from that particular organism. Okay? And then when you look at the next one down in the table itself, you can see there's E. coli there, Escherichia coli. EcoR1 is a very common one that you'll see over and over again in all the examples that we do. But let me show you what ends up happening here. So if you look at its recognition site, you can see, let me just grab a highlighter here. You can see that here, oh, it's not working for me. Oh, because it's a picture, haha, that'll do it. That when we go in this direction, don't copy the arrow, but when we go in that direction, GAATTC, and when we go in that direction, it's also GAATTC. So that's what we mean by palindrome in this context, that the 5' to 3' is identical on both strands, even though they are anti-parallel, okay? So the little arrows that you see here are meant to represent the phosphodiester bonds that are in between those particular bases. Because remember, this is a piece of DNA where we've got them all kind of connected here with their little bonds sticking off here. Okay? So that particular point where you are cutting means that this sequence will be cut between the G and the A on both strands. Between the G and the A on both strands. And keep in mind that keeping them together in here is just hydrogen bonds. So once you cut the main bond holding the strands together, the hydrogen bonds generally tend to fall apart. So you can see it in the next picture how it kind of breaks down. You can see they got overlapping ends there. Those overlapping ends are called sticky ends. And the reason they're called sticky is because I can cut another section of DNA with the exact same type of scissors. I'll get the exact same type of overlapping ends, and I can easily join those two back together because they have complementary hangy bits. So because they have the ability to form little hydrogen bonds that'll hold better together until I have enough time to actually seal up the phosphodiester bonds with ligase, I just buys me a little bit of time. So sticky ends are really nice because they automatically will kind of loosely hold each other until I bind them with new phosphodiesters. Okay. Now the other kind here, you can see it actually forms a line right down through the middle. So when the phosphodiester bonds are opposite each other like this, there's no hydrogen bonds breaking, it's just the phosphodiester bonds. And you can see the result on the side, the GGG-CCC. You can see it splits right down the middle. There's no overhang, so we call this blunt. Blunt end, so sticky end, blunt end. Blunt ends are much harder to stick back together because they don't have that temporary hydrogen bond attraction there. So you have to use a special type of enzyme to put them back together. You'll see that in the next little bit here. Okay, so you get the idea? So when you're looking here at ALU1, cuts right through the middle, sticky or blunt? Obviously blunt. Okay. This one will cut here, and it'll cut here. Now, if I'm going to show the cut, like you will on your test, you're going to show me that it cuts there and that it breaks all these bonds as well. That's what I'm looking for you guys to write on your test for me. Okay. So obviously, what kind of end is that? Okay. And so on and so forth, right? Okay. So again, we got our sticky ends, and we got our blunt ends. Another part of what I'm going to ask you about as we go through is to try and predict, and of course it's all probability, it's all theory, but try and predict how common a specific sequence is going to be? What's the probability that you'll have exactly those 4 bases in a row, or those 5, or those 6 bases in a row? So you're going to want to pay attention to the math that we do here. So first, this is how we do it. We look at something like EcoR1, and we've got this 6 base pair sequence. That's why I've got 4 4s in a row. So 1, 2, 3, 4, 5, 6. And why is there the number 4 there? Because I have 4 options for which bases I could put there. We did that when we looked at the wobble theory earlier. We took how many different possible codons are there and that stuff. So this is the idea that we have A, T, C, and G available at every location. So those are my 4 options. And because I got 6 positions with 4 options, I know that that's 4 to the power of 6 is how we do the probability. 4 options in 6 locations. And so when I multiply that out, I have 1 in 4,096 options. There's 4,096 different versions of a 6-letter sequence that only has 4 letters to it. So this exact sequence for EcoR1 is 1 in 4,096. If I have a smaller base pair setting, so I've got this ALU1, 1, 2, 3, 4, that's why it's 4 to the 4. Well, it's much more common. I would see that exact sequence 1 in 256 nucleotides. So again, that's a theoretical probability. It could be that I've got maybe EcoR1 has the probability of pretty low, 4,096, but maybe it happens 3 or 4 times in a small sequence, or maybe it happens 0 times in a small sequence. It's just a theory thing, okay? But it does help us to kind of figure out how common we think it would be. And for the researcher who's trying to cut a sequence in an exact spot, they want to use the right kind of scissors to do that. So if you want to make lots of cuts, you choose a short palindrome because it'll cut more often. And if you want to make fewer cuts, you use a longer one, maybe a 6, a 7, or an 8 length one, because 8 would be even rarer. Okay. So what is the chance of the recognition site for 8 base pairs? Well, it's 1 in 4 to the 8, because there's 8 positions now. And that, I'm not going to ask you to do the math, I'll just do it for you. But that's 1 in 65,536. So very rare to have that exact sequence available in a DNA sequence. Okay. Just answering the next question, what type of ends does SAL1 produce? Obviously, sticky. And those are the types of questions that you're going to see on your test. So be prepared to answer that kind of stuff, as you can see in your hint there. There is one thing I want to add just before you turn the page, and that's just to clarify up here that we are cutting the phosphodiester bonds. Just so it's clear when you're coming back to study. So there and there, between those bases. Okay, so what's the basic idea? How the actual enzymes do this? Well, quite simple. The restriction endonucleases, or restriction enzymes, are going to look for their recognition site. So they're able to clamp onto the DNA, and they can read. And when they hit that perfect sequence that fits into their jaws, they snip at their active sites. And they're going to have the ability to cut on both strands so that the strands can fall apart for us. And again, we're going to do everything here just like we did when we were breaking and building molecules in the previous unit. All of this gets done with water. And just a reminder that hydrolysis is the opposite of the building. This is the catabolic one where you put the water back in to break the bond. So we add water to break the bond. Hydrolysis, water is bursting something. And it has to be between the right nucleotides. And then because the hydrogen bonds don't have the big phosphodiester bonds holding everything together really nicely, they're pretty easy to make them fall apart because they're just weak little attractions. They're not real bonds, right? They're just a slightly positive hydrogen across from a slightly negative nitrogen, so they're more attracted. Okay, so then we see the same thing here. Here's our little E. coli, this little guy here, and he's moving along in this direction, and then all of a sudden it sees the sequence that it recognizes, and it's able to make its cut. This particular sequence will cut between the G and the A here, and the G and the A on the other side, so then the hydrogen bonds in between will fall apart, and then we get that same sticky thing, sticky end that you see down below there. Okay. I'm just going to flip over now and show you a quick video. So I want you to be able to see these restriction endonucleases in action.
A common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as EcoR1. The enzyme has a precise shape that allows it to run along the groove of the double helix, scanning, in the case of EcoR1, for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, EcoR1 leaves a sticky end. This helps the new gene to attach. The joins are then stitched together by another enzyme called DNA ligase. The genetically engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
So there we're getting the idea then of, sure, this is how the restriction endonucleases are making the cuts, but the purpose of the cuts is that recombinant DNA, to bring in that other piece that's been cut with the exact same scissors so that we can fit them together nicely. They have opposite hydrogen bonds there, sitting there. I'm going to show you this one as well, so you get another way of visualizing it.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, the restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
Okay, so that's
restriction endonucleases.
Calm down. So that's another way to see it. This one I want to show you because it's going to give us the heads up on 3 of our tools. So you've already seen that little DNA ligase keeps coming in and sealing the bonds. It's doing the exact same job as ligase that it did during DNA replication. Its job is always to seal phosphodiester bonds. So once you've made your cut, you're going to be able to obviously seal it back up again. Hopefully your pieces will have joined in together. So one last one before we head back to the note itself.
Using genetic engineering, scientists can create cells that produce proteins they wouldn't ordinarily produce. For example, circular pieces of DNA, called plasmids, found inside bacterial cells, can be engineered to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.
So not a perfect system, but we have ways I'll talk to you about another time about how do we find the plasmid that has the information that we want and then get rid of the ones that we don't want. Because obviously you don't want the plasmid if it doesn't have your info in it, or if it has a double copy of your info, you don't want that. One copy is what you're going for. Right? Okay, so on this page here, I want to give you guys a chance to do a little bit of practice here. So if you go and look on the first page, you'll see what the sequence is for small 1. I want you to read along the sequence like as if you are the restriction enzyme small 1, and make the cut where it's supposed to be made. Draw the line that it's going to need. Okay? So look back at the first page. You can see small 1 is GGGCCC with a blunt end. Questions 1, 2, and 3 are very much so what you'll see on your test. So I'm going to run through and take up the answers with you now. So we already know that small 1 recognizes the sequence CCCGGG, and it cuts in between the C and the G there. So when I'm looking along the sequence, I can say, oh, there's one right there. And if you keep looking, there's a second one right there. Okay? So those are my 2 cuts. Now, the question that's next says, how many fragments will be produced if small 1 digests the sequence? Well, that depends on whether we have linear or circular DNA. What does the question tell us we've been given? Linear DNA. So that's an important piece of information right there, because it means I've got like a string rather than a loop. So I know I've got one here. That end of the string will be cut off. Then they'll have a middle section, and I'll have this third section as well, because it's linear. It's a strand. Right? So I can say quite confidently there's 3 because it's linear. And what type of ends does it produce? Obviously blunt ends. So that's just like one of your test questions. This is also like one of your test questions. So it's a slightly different question, but here's an important piece of information. If EcoR1 was used to cut a 15,000 base pair linear sequence, and that linear is a very important piece of information, then how many cuts would I expect? So we have to go back and see what EcoR1 is. I don't really care what the actual sequence is. I care how many bases in the sequence. So if we go back up and we check EcoR1, it's even at the top of this page here, we can see it's a 6 base pair sequence. So I know that the probability of encountering one has to be that 4 to the 6. So if there's 6 base pairs in the sequence, let me rewrite that because I don't like it. So if there's 6 base pairs in that sequence, then the probability of getting that sequence is 4 to the 6, which was our 4,096. Right? And now I've got to think, okay, if I've got linear DNA, this would be at the beginning, so 0, and I go all the way to 1,500. Okay. I would expect my first cut to be somewhere around the 4,000 mark. 4,096 is the probability. Now, is it actual? Well, it could be anywhere else. But this, again, we're doing probability-wise. What's likely to occur? Then we'd see another one somewhere around 8,000. I'm not really doing a very good scale here, but that's okay. And then I'd see another one somewhere around 12K. I don't care what the exact number is. So about 12K. All right. So if that's the case, and this is a linear piece of DNA, how many fragments do I have? There's 1, 2, 3, 4. 4 pieces, right? But do I really have that full extra piece? Not really. How many cuts do I make was the question, not how many fragments. So how many cuts do I make? I can only make 3. Or again, on average, estimating, theoretically, blah, blah, blah. So here's how I do the math for this, because I don't have to draw a picture every time. So I'm going to say if it's a 15,000 base pair sequence, I'm going to divide that by my probability. My probability was 4 to the 6. So in other words, I'm going to take 15,000 and divide it by 4,096, and I'm going to end up with 3.7 cuts, but you can't have a partial cut. So I'm just going to round that. Do I round it to 4 or 3? I have to round it to whole cuts. So I have to round down to 3, even though the temptation is always to round to the nearest whole number, round to the nearest sensible whole number. I can't make another full cut, therefore 3 cuts. Okay. So how many fragments? Again, we're looking at that picture that I just drew there. So there's 1, 2, 3, 4 fragments. So I expect 4. The question, though, is what if the DNA sequence was circular? Well, here's the theory. If it's linear and you do 3 cuts, 1, 2, 3, no matter where they are, you get 4 pieces, don't you? But if it's circular and you have 3 cuts, how many pieces are there? 3. So that's why I need you to pay attention. Am I giving you linear or am I giving you circular DNA? Because then you'll know it's easy to calculate the cuts, but how many fragments is based on what type of DNA I'm dealing with, linear or circular. Okay? So the next and last one is not something that you would see on your test, a question 4, but it is an exercise, not in futility, but an exercise in trying to pick out palindromes. So I'm going to run through this with you. Remember that palindromes need to be the same in both directions, and they have to be 4 to 8 base pairs long. So you're not looking for a 3 in 2 directions. Here's the thing, though. You can't tell what the other direction is until you write it out. So this is going to take us a little bit of time. But remember that this would be the 5' end. This would be the 3' end. So now we're going to create the complementary DNA underneath, and then we'll go from there. How'd I do, Jordan? It's okay. Don't see any mistakes. Okay, so let's find our first sequence. I'm going to highlight them different colors so that they stand out for us. Where's the first palindrome? Start on the left and work your way over. Minimum 4 bases, same in both directions. The very first 4. GC, GC in this direction, and GC, GC in the other direction. Okay? Keep in mind as well, it's okay if the restriction sites or the recognition sites overlap each other. That's all right as well. So keep moving along. Do you see another one? Yeah, right here, right? You got it. Okay, so that's the 7 that we found in the past. I don't know that there is more than that. There we go. So you get the idea. It's got to be the same in both directions. I'm not going to ask you to do that question number 4 on the test, but questions 1 to 3 are definitely concepts on the test for sure. So I told you at the beginning you want to be asking yourself not just how do these things work, knowing that they came from a biological organism, we stole these from bacteria, but why do bacteria have these in the first place? Why do they need to cut up DNA? And so what they've discovered is that, let me just change my thing here. What they've discovered is that these restriction enzymes are part of the bacteria's immune system. Remember, we're dealing with a single-celled organism without a nucleus. So if it gets infected by a virus, that DNA from the virus has immediate access to every single RNA polymerase that this guy owns because it's not separated by a nucleus. So the bacterial RNA polymerase will immediately begin reading the virus DNA and could potentially be making all these viruses and kill the bacteria very quickly because it builds all these foreign invaders and it explodes when it gets full of the invaders and it spreads, right? So if they have a way to cut up sequences of DNA, then they're able to stop the viral DNA from being effective or being used at all because as soon as it enters the bacterium, it gets chopped up by these scissors. Now, each type of bacteria has a different set of scissors, so they can't cut every sequence known. They can just cut some sequences. So there will be some viruses that are terrible at infecting certain types of bacteria because they've already developed the defense mechanism to fight that virus. But that virus might be wonderful against a different bacteria who doesn't have the right tool. And the idea of these tools is one of those things that can be passed from bacterium to bacterium. Remember we said they're going to share some genetic information with their plasmids? This is one way that we could think about them devising a way to share the wealth on how to defeat the virus, which is pretty cool. Okay, so it's part of their original immune system. Remember that the type of viruses that will attack a bacterium are called bacteriophages. So that's what they would be used to destroy is the viral DNA. Okay, and it's cleaved before the viral DNA can be used. So on this next page here, just an example of it. There's a bacteriophage that is injecting its DNA into the bacterium. You can see bacterial chromosomes just sitting there freely in the nucleoid region. And of course, you don't want that DNA to get its hold on the machinery there. So here's the restriction enzyme just comes up, chops it. It's useless, can't be read. It gets recycled that way. Okay. So we know how to cut the sequence. In those little videos I already showed you, we know how to join it back together again. And that's with our DNA ligase. Okay? And it will create a phosphodiester bond, and it's between the bases, but it's on the sugar phosphate backbone. That's where the actual bond exists. It doesn't join the two bases themselves, it joins their sugars and their phosphates together. Okay. And of course, how do we do that? Well, if we add a water to break the bond through hydrolysis, then of course we're going to remove a water to join the bond through dehydration synthesis, which of course is the same thing as condensation. So this is our dehydration synthesis reaction. I've already mentioned this, but sticky ends are so much more useful because you get to join two DNA molecules together, but they already have that loose little hydrogen bond already forming, and it gives the DNA ligase enough time to get there and seal those phosphodiester bonds to make it a permanent join, as long as they're complementary sticky ends. But if you have a blunt end, you can't use your normal ligase like you would in any other cell. So instead, the researchers have figured out that this T4 version of the DNA ligase, T4 meaning from a T4 bacteriophage, which is a viral enzyme that they've stolen. And they can use that to put the sticky ends together. Sorry, I'm lying to you. They can use that to put the blunt ends together because they don't have any hydrogen bonds holding them together. So you have to hold both strands and seal at the same time. It's kind of the idea. Okay. So here again is a picture of a sticky end being shown here. We got the DNA ligase coming along now, And instead of breaking bonds, we've got these little hydrogen bonds here that have already begun to form between the sticky ones. So we have our H bonds in between here. And it's the phosphodiester bonds that we're sealing up. So here we've got phosphodiester bond. And of course, that's happening on both strands. So the exact point that the restriction endonuclease would have cut. So the very next question you should be thinking of is, okay, if these are bacterial enzymes that do the cutting, because we want to cut foreign pieces of DNA, how then do the bacteria not just end up chopping up their own DNA? They must be doing something to protect theirs from their own enzymes. And in fact, what ends up happening is they know how to distinguish or tell the difference between their DNA and outsider DNA because they have these enzymes called methylases. Methylase is the enzyme that will add a methyl group. And it adds that methyl group to the recognition site on its own strand of DNA. So you can see that we've got the CH3 here that's been added to the recognition site. That is called a methyl group. You see them on both strands. So if this is an E. coli piece of DNA, belongs to the bacterium itself, it doesn't want its own EcoR1 to cut it, so you block the recognition site for your own enzyme. And so you're only ever cutting up foreign DNA at that exact recognition site. Pretty simple. Any unmethylated is fair game. So if they forget to methylate their own, it's fair game. The enzymes will cut as soon as they bump into that sequence. Okay? So that's what the bacterium will do. And once again, we steal their good idea and we say, well, that's a great idea. Maybe in the sequence that I'm trying to move around, I know that there's the EcoR1 site in the middle of my gene and I don't want to cut that. So I can methylate as well. And then my whole gene will be transferred properly when I'm doing this recombinant DNA stuff. So I can also protect sequences. So maybe I protect the EcoR1 site so it never gets cut, and then I can use at least at that one location, and then maybe it cuts it somewhere else, but at least it's protected for that moment. Okay? So the next part of our session we'll be talking about tools that we have created on our own. We haven't stolen from bacteria, and one of them is this gel electrophoresis. We won't have enough time to finish this conversation in this lesson, but we will pick it up on the next lesson. So let's get started on it anyway. Okay, here's the basic idea. We've got this little tub. It's got some solution in it. It's got on each end, you see there's an electrode, so negative and a positive end that's plugged into a power supply. In the middle there, you see something called a gel. It's usually made of agarose. Agarose is a type of protein. And built into the agarose, or what agarose is just long kind of chains, kind of like fibers. And they're all kind of like a mesh, all mushed in together. Kind of like if you've ever watched Survivor and you see anytime they have to run through a rope course. But the rope course has just the ropes in all different directions, right? Imagine that you are a group of 20 people having to hold hands to climb through that rope course. I think you can probably imagine it would take you a lot longer as the group of 20 than the group of 2 to make it to the end. And that's the same principle here. This agarose gel with all of its mesh and all the ropes in the way, if you are a short fragment of DNA, you will be able to maneuver much quicker and much farther through the gel, through the obstacle course, than a long piece of DNA. So what this will do, this little machine will allow us to separate fragments of DNA based on their lengths. One of the questions on your test is going to be asking about these tools, the 3 we've already mentioned, and this one here, and a few going forward. I'm going to ask you, basically, what's in a few words, just in one sentence, what do biologists use these tools for? So if you choose gel electrophoresis, the very simple answer is it's used to separate DNA based on their lengths. And then you kind of have to have some idea of how it works, but that's the basic short answer that I'd want for that. We can also use this for proteins as well. Bigger proteins obviously won't travel as far as small proteins will be able to travel, or polypeptides. We can, different chain lengths, we can always separate this way. So the key to getting it to move through and not just sit here in this gel, what we'll end up doing is if you can see these little boxes here, these little boxes are where there is actually no gel. It forms just like a little hole, and that's called a well. So these are the wells, and that's where you put your sample of DNA in there. So DNA gets put here. Okay. Now DNA, this is a key idea, is negatively charged. Okay, and that's because we have all those phosphate groups sitting along the side of the ladder with all the sugars there. Okay, and the phosphates have lots of angry oxygens that aren't fully satisfied, therefore they're overall negatively charged, unhappy little things. So we have a negatively charged molecule, and as soon as we flip on the power, you can see we've got this negative electrode on this side and a positive electrode on this side. So the DNA is going to begin to migrate from the negative end to the positive end to try and get away from the negative and try to move towards the positive. It's drawn. So the DNA will immediately begin the race, but then the size of the DNA will determine how far it gets before I turn off the power. As soon as I turn off the power, everybody stops the race. Okay? In that case, then, we're going to have short fragments. Well, I'm not meaning that. I'm meaning that as an arrow for direction of pathway, not the length of the strand. So short fragments are going to travel really far, and then long fragments are going to get stuck in the mesh towards the beginning. So long fragments move slowly. And that's where we'll leave it for today.